Hello, hello. Welcome to my video, Transcription, Translation, and the Consequences of Mutation. It's like a little rhyme. So the goal of this video is to try and help you understand the material of week 5, to give you the overall perspective, as well as to get down to the details of how these processes work. So hopefully the homework will make more sense and you'll be able to grasp the concepts really well and do well on the exam as well. If you have any questions, please feel free to reach out to me. But hopefully we will cover everything in this video and it will make sense to you. Okay, let's see what we can do here. So this week, what we're focusing on is how we can actually put the genes to use. So far in this course, we've talked about genes on the DNA. We've spent all of this time about DNA and the genes. And we told you that the genes were recipes on how to build proteins, recipes for building proteins. And proteins are really, really important. It turns out there are four major categories of biological molecules used by cells. There's fats, and you don't really need to know this part here. So this is just to give you an idea of what we're doing here. Fats, their job is energy and buffer. Their job is they're the wall. They're what make up the wall of your cell. So basically they're for energy and for being a wall. So really, wall and energy. But basically their job is to just sit there. They don't do anything. They just sit there and by the nature of their existence happen to do their job. Sugars are the second category, or carbohydrates is the more specific term. They are for energy and sometimes for signaling, but that's pretty much their only job. Nucleic acids are the third category, acids. And these are DNA and RNA. So that's DNA and RNA, which we'll learn about RNA this week. That's DNA and RNA, and their whole job is information storage. So so they don't actually do anything. Actually, there's one or two exceptions to that, but they don't actually do anything. Their job is to just sit there and contain information. So notice these guys don't do anything. These guys don't actually do anything, and these guys don't actually do anything either. So there's no action happening here. Everybody's just sitting there and doing their job by the nature of simply sitting there. But cells are very active things. They're living, they're alive, they do jobs, they move, they function. And so that means our fourth category of molecule has to do everything. I know, it's like being a parent, right? The kids don't do anything, so you have to do everything. The fourth category is proteins. And proteins are essentially the doers. They do everything. And unlike nucleic acids, where every DNA strand is going to look the same, right? Maybe we changed up the sequence, right? But DNA is always going to be double-stranded. It's always going to be sitting there. Each DNA strand is essentially very, very much the same, with the exception of changing the sequence. Proteins, we change them all, depending on the sequence and a bunch of these things that we'll look into. Depending on how you design the protein, you can completely change what it does. So these things, proteins, are essentially what make you up. And that's why we say a gene, even though they're recipes for only one of these four molecules, because they build proteins, and proteins are the most essential, are the most active dynamic part of the cell. By coding for proteins, that's how DNA controls how you turn out. Because proteins are what color your skin. There's a protein called melanin that makes your skin darker or lighter depending on how much of it you have. It's the one, or proteins change your eye color, proteins change your hair color, proteins affect what diseases you have. Proteins do everything. And so because they do everything, depending on the versions of these proteins you get, entirely depends on what your cells do and how they react to situations. So proteins are very, very important, which is why the genes that we've been learning about are very, very important, because they have the instructions on how to build the things that do everything in the cell. So, and this week we're going to learn how we read this message and build the protein. We've learned that that's what they do. This week we're going to learn how, and hopefully I'll be able to show you why as well. But hopefully that gives you an idea of why this is so important. Proteins do everything. So the whole goal of everything we do this week is to take the instructions that we've been keeping in the DNA this whole time and build proteins out of them. Okay? All right. All right, here we go. So we talked about why proteins are important and that they do everything in a cell. Let's give you a bit more background on specifically some things that proteins do, and then we'll go into the process of how we build them. So proteins do a lot of different things, and because they do so many different things, there are different categories of proteins that address different needs of the cell. And there's tons of these, just a few major categories, like you could have enzymes. So there are enzymes, there are signal molecules, which are also known as hormones, usually, though not always. So those could be hormones, or there are other kinds of signal proteins. You can have receptors, contractile molecules, defending molecules, all kinds of different proteins. Structural. There's another one. Proteins really do all kinds of things. Transport, there's another one, transport. So like enzymes, we've learned about enzymes before. Enzymes are proteins that make chemical reactions happen faster. So for example, DNA polymerase 3 that we met in week 3, that is an enzyme. Let me change colors here. So DNA polymerase 3, that was an enzyme protein because it made a chemical reaction happen faster. It took DNA nucleotides and linked them all together. So DNA polymerase 3 is an example of an enzyme. Helicase is an example of an enzyme. The enzymes we learned about this week, RNA polymerase 2, those are all enzymes. So proteins that make reactions happen are enzymes. There are signal proteins. They can be hormones, or they can just be poking off the side of the cell. But regardless, their job is to send a message from one cell to a different cell. So an example of because sometimes, because cells in your body are specialized, right? A nerve cell is different from like a muscle, or from like a reproductive cell, for example, or something. Like you need the brain cell has to be able to tell the other cells in the body what to do to respond to a certain situation. They have to get them to act. So like if I want to try and get a cell to divide, or if I want to signal for a cell to go through meiosis, for example, I need to send a little molecule through the bloodstream that's basically like putting a letter in the mail, shipping it off to that cell, and then the cell has to receive that message and act on it. So these signal molecules, the hormones, are proteins that are the letters. So one cell builds it and drops it off in the bloodstream, and the other one picks it up, and that's the signal to do this. So an example of that is insulin. Switch back to blue here. Oh, yeah. Is insulin. Oh man, I am struggling today, guys. I am sorry. Insulin. You've probably heard of insulin because it's associated with diabetes and stuff. Insulin is a protein that is manufactured by cells in the pancreas and released into the blood. And the signal that insulin sends to cells that receive it is there is sugar out in the blood, bring it into your cell and burn it for energy. Because that's the whole point of sugar, is for your cells to burn it for energy, but they have to, the sugar doesn't just naturally go inside a cell. A cell has to know there's sugar out there to bring it in and burn it. And so the pancreas signals or builds these little insulin proteins and drops them into the bloodstream so that cells everywhere can receive that message and know to bring in the sugar. So when diabetics don't have insulin, then that's why their blood sugar gets so high, because a healthy person that doesn't have diabetes, their body detects that there's sugar, so their blood sugar starts rising because their digestive system's pulling sugar out, whatever they ate, it's rising, and so then it drops insulin in there, and the cells start pulling it out, so it maintains it at a certain level, because the cells are pulling it out and burning it for energy. So we're pulling out as much as we're putting into the bloodstream. But a diabetic, because their problem is their cells aren't producing insulin, or at least a type 1 diabetic, their cells aren't producing insulin. And so even though there's sugar out there, the cells aren't getting the message. And so nobody's taking the sugar out of the bloodstream. And so that's why their blood sugar goes super high, because all of the sugar you're eating is just going straight into the blood and not being pulled back out again. So these signal hormones are very, 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 very important in the body. We won't really focus as much on them. Most of the proteins in this class that we learn about will be enzymes. These other ones are just examples to show you all of the different kinds of things a protein can do in the cell. Receptors, we talked about these signal molecules, how somebody drops them in the mail. You need a mailbox in order to receive the letter. So a receptor is like a mailbox. Its job is to pick up the hormone, pick up the signal molecule, and tell the cell to do something. So it picks it up, reads it, and says, aha, and now we've got to go act on that. So insulin has a receptor, and it's called insulin receptor. We're super clever about that. Insulin receptor. Again, these are just examples so that you've got a general idea. So its job is to grab the message of insulin out of the blood and be like, aha, it's time to bring the sugar in, and tells the cell to do that. Contractile molecules, in order to get your muscle cells to flex, they have a protein called myosin. I'll write that one down. They have a protein called myosin that its whole job in life is to grab onto ropes. They're basically like they're a lot like the spindle fibers that we used in mitosis. They're just these little protein ropes throughout the cell. And it grabs onto those ropes and pull. That's all it does. And by doing that, we basically play tug of war and drag the edges of the cell closer, and that's how you flex your muscle. There's proteins that do that. Myosin is the special one whose job it is to flex your muscles. So there's a contractile one. Defensive, your immune system is savage And it has to be able to take down bacteria. Well, it's got proteins like there's one called lysozyme. Lysozyme is an enzyme, so notice it can be in multiple categories, and proteins can be in more than one category at a time. Lysozyme is an enzyme that the cells spit at an invading bacteria cell, and it goes in there and eats their little armored wall around the outside, and the bacteria cells literally explode. It's a good way to kill a bacteria, right? And make them explode. The immune system loves popping things and spilling their guts out and blowing things up. It's all sorts of great fun. So there are defensive proteins that are used to either protect cells or attack other cells, depending on your particular function. The structural proteins, cells usually need to maintain a specific shape, right? Cells, we told you that the outer barrier is just fat, and fat loves to be in a circle. If you've ever dropped like a droplet of oil in water, it likes to be in circles. Sometimes a cell doesn't want to be in a circle, right? I need to be able to move. I need to be able to eat things. I need to be able to do all kinds of things that make me not want to be in a circle. And so I need to have proteins like those spindle fibers that we used in mitosis and meiosis. We use proteins that make big long strings, big long poles that I can arrange in a certain way inside the cell to help arrange my shape, or to pull chromosomes apart, or to do things like that. So an example of that would be the spindle fibers. And then transport, when we got that signal from insulin, we needed to have a protein that's going to grab the sugar on the outside and bring it inside. So we've got all kinds of transport proteins. There's a protein called aquaporin. Aquaporin. And its job is, it's basically just a pipe that a cell sticks in the wall to allow water to go in and out. It's a transport protein. So these are just a few examples of all of the different kinds of proteins that you can do. You've got thousands, there's hundreds of thousands. We haven't even discovered all of the proteins that are possible, that are in all the different species and stuff. There are tons of proteins, but hopefully this gives you kind of an idea of all of the different kinds of things, of a bunch of the different kinds of things a protein can do. So now let's finally get down to the business of how we, to the business of this video, namely, how do we build said helpful proteins? In the DNA, we have instructions on how to build them, basically a recipe on how to build them right. So the DNA's got the instructions. So normally we want to, so basically this week we're studying the process of how you go from DNA to protein. And this overall process here is called central dogma. That's a good vocab word to know. Central dogma is the overall process of how to get from DNA to protein. How do we read the instructions of DNA and build a protein out of it? That whole process is called central dogma. Now, there are several steps in the middle or that all work together, that all add up to central dogma. So central dogma is the general idea of getting from DNA to protein. And then there are sub-steps, sub-processes to central dogma. Namely, the two that we focus on in this class specifically are called transcription and translation. So these two steps together make up central dogma. So between the two of these, we use these two steps to get from DNA to protein, and together transcription and translation make up central dogma. So that's a very good vocab word to know, and to be able to differentiate between the whole overall process, which is central dogma, and the specific steps of the process, which are transcription and translation. Okay, so now let's work, because most people think like, well, if the DNA's got the instructions, then why don't we just read the DNA and build a protein? Why do we have a two-step process here? And that's because in reality, central dogma doesn't just go straight from DNA to protein. There is a middle step here. We go from DNA to RNA to protein. And people often wonder, like, well, why do we go to RNA and then to protein? Well, here's one reason. Inside your cell, let's pretend this rectangle here is my cell here. We have this little bubble inside the cell called a nucleus. And the DNA is kept inside the nucleus. This is where we hide our chromosomes. So your 46 chromosomes are kept in here. So this is where the DNA is. Now, it's important that we keep it in the nucleus, at least in human cells. It's important that we keep the DNA inside the nucleus because that's where we keep it safe. It's like putting plans for a building inside a filing cabinet or money inside a vault. It's in a protected case all by itself where outside factors have a hard time damaging it. Because if I damage the original instructions, you know, bad things happen because I can't really fix them very easily, especially depending on the kind of damage that I do to them. So I want to keep them inside their little protective bubble here so that they don't get damaged. Specifically, the cell has these little defensive enzymes out here called endonucleases and exonucleases, and their job is to chew up DNA. That's their whole job in life. It's actually a virus protection mechanism. Their whole job is to find any DNA that they bump into out here and rip it to pieces. And turns out that if you have your chromosomes ripped to pieces, you can't survive. So we need to keep the defensive mechanisms outside, away from the DNA, so that they only rip aside, or so that they only rip apart the things that I need to protect from. Because like viruses, for example, viruses work by injecting their DNA into the cell and commandeering your central dogma process to build new baby viruses. So any DNA that is outside the nucleus is really a bad guy. And so I want this guy to come in here and rip that apart. But he can't tell the difference between friend and foe. So he'll just destroy any DNA he meets. So we want to keep the exonucleases out here and the DNA in here where it's safe and they can't get to each other. But the problem is the ribosomes. A ribosome is the enzyme. A ribosome is the enzyme responsible for building the protein. This is the builder, the protein builder. But the problem is the ribosomes live out here. So essentially what we've got is kind of a little predicament, because these guys are actually too big to get into the nucleus. And I don't want the DNA to leave the nucleus because it could be destroyed. So we've got this kind of predicament here where I've got the plans at the architect's office and the construction workers are at the construction site, but I don't want the plans to leave the architect's office because they could get destroyed. But these guys need the plans in order to build a building. So we've got this distance in between the two of them that we need to be able to surmount while keeping everybody happy. I need the construction workers to get instructions without having the original copy of the instructions leave the nucleus. So we solved that by photocopying the instructions. If I can make a cheap photocopy of the original plans for the building and then send that to the construction site, I can keep the originals back at the architect's office, send the copy off to the construction workers, and they'll work on it. And if it gets rained on or the wind blows it away or it gets torn or dirt on it or something, the construction workers can just call me and I can photocopy the originals again and send them a new copy. So by photocopying the DNA photocopying the gene and then sending it out to the ribosomes, I'm able to send them a copy that I'm not too worried about getting destroyed while keeping the originals inside the cell. That's why we do two different steps. So that the DNA can stay where it is, the ribosomes can stay where they are, and everyone still gets their jobs done. So we have a process where we photocopy the DNA into RNA. And the reason why we use RNA is it's simply cheaper for the cell to use. It's single-stranded, so I only have to build one strand instead of two. The nucleotides are easier to make. It's just overall cheaper for the cell to build in RNA. The problem is RNA just doesn't last very long. It's very prone to being damaged and stuff. It maybe lasts 5 minutes. Now, I don't want the DNA is much more stable. It can last for several decades, centuries even. You learn from King Tut that we can pull DNA out of mummies that died 5,000 years ago. So DNA is very, very stable. RNA lasts maybe 5 minutes, but it's a good, cheap molecule for me to use to simply send a message, and then I can photocopy again later. So we copy into RNA simply because it's cheaper to build than DNA is. So we photocopy it into RNA, and then we'll take the RNA outside the nucleus and give it to the ribosomes. And then the ribosomes are going to do the second step here to build a protein. And I'll draw my little protein over here. There's my protein. Okay, so we got a two-step process here, and in the middle, I'm moving the instructions outside of the nucleus to the ribosome. So this first process here, process number 1, is called transcription. And so transcription is where we photocopy the DNA. We're going to read the DNA sequence and build a photocopy of it in RNA. So let's walk through how that works. We'll start with transcription, then we'll come back for that second step. So here's my little simulator. Hopefully this will help demonstrate how this process works. We're going to focus on transcription here, so we're just focusing on the top. What I have is double-stranded DNA right here. So this is the DNA. This is like the gene that I'm looking at that's got the instructions for how to build a certain protein. And so we've got 5' and 3', 5' and 3'. Now it turns out that DNA, contrary to what most people believe, DNA is double-stranded, but only one of the two strands actually contains the instructions, actually contains the gene. The other strand is basically worthless. We count it as part of the genetic region because, yes, it sticks to the gene, but essentially we don't care about the other strand. We only care about the one strand that has the template, or that has the gene on it. So the way that I like to think about this is artists, you know, when an artist does a nice charcoal drawing or something, the drawing is what people really want to look at, right? They want to look at the drawing, but often the artist takes another piece of just blank paper and sets it on top of the drawing in order to keep the drawing from getting smudged or damaged or ruined or faded or something like that. So one of the strands is like the picture. It actually has the instructions, and the other strand is the blank sheet of paper that's simply there to keep it protected and happy and safe. So the strand that actually has the gene on it, the strand that we're going to read, is called the template strand. Okay, so the strand we're actually going to read is called the template strand, because a template is something that you trace, right? If you have a template for one of your classes, it's a standard format that you follow. Or if you have a template in drawing, it's usually something that you trace. The template strand is the one we're going to trace. That's the one we're going to read in order to build the RNA. The non-template strand is the one we ignore, right? We're going to separate the DNA strands and completely ignore the non-template strand. It has absolutely no job here whatsoever, other than being the second strand in double-stranded DNA. So make sure you notice that you know that difference. The template strand is the one we're actually going to read in transcription. The non-template strand we completely ignore. Okay? All right. So in DNA replication, right, in order to get to the message in DNA replication, we had to separate the two strands and then have a primer come in and be built, right? Helicase separated the strands, a primer had to be built. DNA polymerase had to come in and build it. You're probably like, oh no, I already took exam 1. Never talk about that stuff ever again. Like, sorry, it comes back to haunt you here. So we had like 8 different proteins that were involved in DNA replication. Well, the nice thing about transcription is there's only one. RNA polymerase, so its name is RNA polymerase 2, as opposed to DNA polymerase 3. So its enzyme name is RNA polymerase 2. Sometimes it's abbreviated RNA pol, but its full name is RNA polymerase 2. Its job, it is the one-man band that does transcription. That's it. So RNA polymerase 2 separates the two strands. RNA polymerase 2 reads the template strand. RNA polymerase 2 builds the RNA. It's a one-man band. So RNA polymerase 2 does everything in transcription. So let's come in here. So RNA polymerase 2 comes in and hops on the DNA at a spot called the transcription start site. So remember, in DNA replication, we started unwinding the DNA at the origin of replication, right? So that was where we started, and then we put on a primer and we built. In this case, we start transcription at a spot called the transcription start site because we're so creative. And there are ways to tell where the transcription start site is. There are sequences in the regulatory region that help cells determine where the transcription start site is. In this class, we don't expect you to know that. So in this class, we will simply tell you where the transcription start site is, and you're just going to have to believe us. So the transcription start site is literally start photocopying here. Okay. And there is a transcription stop site that does the exact same thing. It says stop building here. So we have a start and a stop. In this case, I'm going to say the stop is right over here, though usually your mRNAs are really, really long. Usually your RNA molecules are really long. So we have a transcription start site and a transcription stop site. And that's what we call them. Transcription start site, transcription stop site, because we're very creative. RNA polymerase 2, unlike DNA polymerase 3, it doesn't need a primer. So we can just start at the transcription start site and just start building. So we hop on at the transcription start site. Get my mouse back here. Okay, here we go. We start at the transcription start site and we separate the two strands. So I'll take my template strand here, which is the bottom one in this case, but remember, it's not always. We will always tell you. That's the other thing. The cell has the ability to tell the template strand apart from the non-template strand. There are special sequences, again, beforehand that tell it which one's the template strand and which one's the non-template strand. In this class, we don't expect you to know how it figures that out. We will always tell you which of the two strands is the template strand and which one is the non-template strand. And it may not always be the bottom strand. Right? Top and bottom strand is completely arbitrary terms, right? We can flip it. It just depends on whether you're standing on your head or standing on your feet. I didn't change the DNA. So just always make sure to look which strand is the non-template strand and which one's the template strand. In my particular example, I put the template strand on the bottom, but that's not necessarily always the case. All right. So we separate the two strands and we completely ignore the non-template strand. It's as if it doesn't exist. Okay, and we're just going to come down here to the template strand. And then RNA polymerase 2 is going to come in at the transcription start site, and it conveniently builds in the 5' to 3' direction, just like we did with DNA polymerase 3. You always build in the 5' to 3' direction. So we're going to start at the transcription start site, and then RNA is very similar to DNA. we're going to build in the 5' to 3' direction. So there is a, so like here, C pairs with G, C pairs with G, G pairs with C, T pairs with A. Now A, here's your catch. And this is where I'm going to pause and go look at the differences between DNA and RNA, because this is one of the main differences. In RNA, C pairs with G and A pairs with U. We add a new nucleotide this week. It's called uracil. And the letter for that is U. So A pairs with U and C pairs with G in RNA. So let's go look at the differences between DNA and RNA. There are 3. 1, RNA is single-stranded, DNA is double-stranded. That one's pretty basic. The second one is look at the sugars. On the sugars, so DNA is on the left, RNA is on the right. This is just your basic structure, right? You remember there's the phosphate, the sugar, and the nitrogenous base. So there's week 2. You're like, oh no, not again. So the phosphate, the sugar, and the nitrogenous base. If you look at it, this little OH, that's the 3' end. So that's the OH that we stick the phosphate to build the strand. On DNA, there's just an OH right here on the 3'. On RNA, there's actually also an OH on the 2' carbon here. So it has 2 OHs instead of 1. That's actually where the name comes from, because this pentagon over here, the pentagon is the sugar. This sugar over here, this is called ribose. That's the kind of sugar, just like glucose is a different kind of sugar. Ribose is a sugar. That's the kind of sugar the pentagon is in RNA. So ribose has these 2 oxygens poking off the bottom here. Notice over here on the DNA side, we have the pentagon, same pentagon, except we only have 1 of the 2 oxygens. So we're actually a ribose that's missing an oxygen. So over here, this was called ribose. So over here, this one's called a ribose that's missing an oxygen, or deoxyribose. Hence the name of this one is deoxyribose nucleic acid, or DNA. And this one over here is ribose nucleic acid, or RNA. So there you go. The difference in the sugars is built into the name. The third difference is the one that we were talking about over here on this one. RNA does not use thymine. It uses uracil instead as its 4th base. And the reasoning for that basically is uracil is cheaper to make than thymine. And remember, our RNA is supposed to be the cheap paper. So we're simply saving money by using uracil instead of thymine when building an RNA. But notice this is the trippy part, because here, notice thymine paired with adenine, but over here I have an adenine pairing to a uracil. So you have to be able to swap back and forth between the 2 languages, because up here in DNA, basically in RNA, you're just going to take everywhere you would have put a T and stick a U there instead. So T still pairs with A, but then when you're talking about RNA, A pairs with U. So here, C pairs with G, C pairs with G. I'll continue my transcription here. C pairs with G, C pairs with G, G pairs with C, T pairs with A pairs with U, T pairs with A, and then C, G, C, oops, C, U, G, C, A, because A pairs with T, U, G, C, A, U, C, G, G, A, U, C, U, A, U, U, G, C, U, A, G, C, U. And then if that were my transcription stop site, I would stop transcription. RNA polymerase 2 would let go of the DNA. The DNA would reform. So let's pretend that was my stop site over here. This is the 3' end now. Dash 3'. Okay, okay. Sorry, I'm working in Excel here. Okay, we'll make it happen. There we go. So now the DNA sticks back together again with the non-template strand. So the template strand comes back up here, boop, and we're done. Now I just created a single-stranded piece of RNA that's a photocopy of the DNA. Now notice it's complementary to the template strand. So a lot of people get this mixed up because they say, well, it's just the non-template strand. But remember, we totally ignore the non-template strand. But if you look at the sequence, with the exception of U's and T's, it's the exact same sequence as the non-template strand. But that's not because we used the non-template strand in transcription. We completely ignored the non-template strand. The reason why their sequences are the same is both of them are complementary to the template strand. That's why they're the same. We're not reading the non-template strand. We're completely ignoring it. We read the template strand to build a complementary RNA strand that happens to be the same sequence as the non-template strand. Okay, so don't get that confused. We read the template strand, not the non-template strand. But because both the RNA and the non-template strand are complementary to the template strand, their sequence will be the same, except you swap out the T's for U's. Okay, so this is an RNA. Now we put letters in front of the RNA to tell what job it's doing. There are lots of different jobs done by RNA. And so we put a little letter in front of the name RNA to say what specific job this particular RNA is doing. This particular RNA, its job is to carry the message of the DNA out to the construction workers, the ribosomes. So this particular kind of RNA, the one that has the gene on it, is called mRNA. We'll write that out over here. mRNA, not in caps, sorry, mRNA, which stands for messenger, because that's its job. It's an RNA molecule that's behaving as a messenger. It's carrying the message of the gene from inside the nucleus to outside the nucleus. So it's called mRNA, or messenger RNA. Okay, so that's the messenger RNA. Now that's transcription. That's all it was. We read the template strand, we build a complementary RNA strand in the 5' to 3' direction. The DNA seals back together, and ta-da, I have a single-stranded piece of RNA. That's it. That's transcription. That's our first step. Okay, now let's come back to our little diagram over here. We've turned it into RNA. Now we have to take RNA. The RNA will be carried outside. That's not really an extra step, but the RNA needs to be carried outside the nucleus to the construction workers, the ribosomes out here, and the ribosomes are going to do our second step. So this step over here, that's our second step. So that was our first step. There's our second step. The second step is translation. And the translation is the process of going, so transcription was going from DNA to RNA. Translation is going from RNA to protein. Specifically, it's actually going from RNA to polypeptides, which I'll talk about here in a sec. But in a general idea, for right now, we'll write about how it goes from RNA to protein, okay? Okay, so that's translation, is going from RNA to protein. Transcription is going from DNA to RNA. Translation is RNA to protein. Sometimes people get those names backwards. Make sure that you know that transcription happens first, translation happens second. Okay, let's go over how you do translation, and then I'll talk about ways you can remember which one's which. Because that can be tricky too. I've got some little mnemonics and things you can use to remember that. Okay, so let's pop back to my little simulator here. Okay, so we're going to take the mRNA strand, this one right here, We're going to carry it out of the nucleus down to my little handy dandy ribosome friend down here. We carry it down here. Okay, so now we're outside of the nucleus. We're in what's called the cytoplasm, which is basically just the water inside the cell. We're just inside the cell now rather than inside the nucleus. So we're out here and we've come to the ribosome. Now the ribosome is a very interesting enzyme. It's freaking huge. It's a massive, massive enzyme, and it's made of actually a ton of proteins all stuck together, working together to do a job. So it's one massive enzyme made up of a ton of different pieces of protein. And it's actually a novelty in that it's about 2/3 of it by weight is also made of RNA. So it's an enzyme that is half made of RNA, half made of protein, which you don't see very often in the cell at all. But the RNA that is used in the ribosome is not mRNA because it's not doing the job of carrying a gene from inside the cell to outside the cell. It's doing the job of being part of the ribosome. So we call it ribosomal RNA or rRNA. So there's the second kind of RNA that we ask you to learn for this week. mRNA carried the message out to the ribosomes, the message of the gene. rRNA is part of the ribosome. Okay, so yeah, these ribosomes are huge. And basically, they're made of two general pieces that bite around the RNA like a sandwich. So like the RNA, the mRNA is like the meat in the middle, and then we've got a big piece of bread on top and a big piece of bread on the bottom, and then clamp down around it. And that's how we read the mRNA. It's a really, really cool process. In my little simplified version, I'm kind of showing this in cross section here. Normally, the mRNA would go right through the middle of this big blob. But just bear with me. The ribosome is kind of taking up this whole area right here. But I just wanted to put the label up here because I'm going to need this space in the middle here. But just know the ribosome is grabbing onto the mRNA. Now notice the ribosome is grabbing on at the 5' end. And this is where it throws people off because we all, in past weeks, especially with transcription and with DNA replication, you always read 3 to 5. You build 5 to 3. Proteins aren't made out of nucleotides, so they don't have a 5' to 3' end. So proteins are not limited by having to be built in the 5' to 3' direction because they don't have a 5' and a 3'. So we actually read the mRNA in the 5' to 3' direction and build the protein that way. Right? We don't have a 5' to 3' direction. In reality, we've got different terms for proteins, but that you don't need to know. So just remember, you read the mRNA in the 5' to 3' direction instead of the 3' to 5' like we had to do with transcription in the other one. So the message is read in the 5' to 3' direction here in order to build the protein. Okay? So we're starting at the 5' end. Now, we started transcription at the transcription start site. Translation, which is this second step of reading the mRNA and building the protein, does not start at the transcription start site, which means it doesn't start at the beginning of the mRNA. It starts at a particular code, which I'll show you here in a sec. It turns out that, actually, yeah, this is a really good place to talk about this. Transcription, the way that I like to think about transcription is we're going from one language to a photocopy of the same language. Like in the old days, before we had printing presses and whatnot, in order to take a book, like maybe the scriptures or something, a monk or somebody had to take a scroll and sit down, or a scroll with the scripture on it, and then sit down with another scroll, piece of paper that was blank, and copy it word for word, right? And the process of going from that one to that one was called transcribing. That's where the term, a scribe comes from. So I'm reading English and writing English. It's the same language, we're just changing papers. DNA and RNA are both made out of nucleotides. So I'm just going from one language and copying it into the other, hence the name transcription. There's a way that you can remember that transcription is going from DNA to RNA because we're staying in the same language. I'm just photocopying it. Translation is different because proteins are not made out of nucleotides. They're made out of something completely different, which is they're made out of amino acids. Oops. They're made out of amino acids, and amino acids are nothing like a nucleotide, nothing at all. I'll just show you some pictures over here. These are kind of what your nucleotides look like. Let me show you. This is more of the specific chemical structure of what a DNA nucleotide looks like. Compare it to, here's an example of an amino acid. There's a different one. There's another one. There's another one. One more. Notice they are totally different molecules. There is no similarity whatsoever between a nucleotide and an amino acid. So amino acids are the building blocks of proteins, where nucleotides were the building blocks of DNA and RNA. But there are so many differences between protein language and RNA language. RNA language is written in 4 letters, A, C, G, and U. DNA language is written in 20 letters. There are 20 different amino acids. So we completely have to jump languages, which the term for jumping languages is translation. We're reading English and turning it into German. Totally different sentence structure, totally different everything. So we have to translate. So in order to translate from the RNA to the protein, there's a code that we use that says this word in RNA means that letter in protein. And the RNA, in this code, the RNA is read in groups of 3 letters at a time. So 3 letters spells one RNA word. And then the RNA is basically a sentence of one 3-letter word after another. And then the ribosome basically reads the 3-letter code on the RNA, on the mRNA, and knows which amino acid to put in that spot. And I'll show you how it does that. So the amino acids, they've got these little short, or these names here. All of them have crazy names, from simple names like alanine to a really fun one called phenylalanine or tryptophan or glutamine or aspartic acid. You don't need to know them. We are not going to make you memorize the 20 amino acids, which is why we don't have you write them out. Amino acids, we usually abbreviate them into 3 letters. So each amino acid has a little 3-letter code. So in my example here, the amino acids are these little beige pieces. So that's glutamic acid, tryptophan, cysteine, methionine, histine, etc. So each amino acid is going to correspond to a trio of letters on the mRNA. The trio of letters in the mRNA is what is referred to as a codon. A codon is the trio of letters on the mRNA that translates to an amino acid on the protein. That's what a codon is. The trio of letters on, the trio of letters of mRNA letters that translates to a specific amino acid. Okay, Excel. We love technology, especially when it doesn't work for us, right? Okay, a codon is the trio of mRNA letters that translates to a specific amino acid. It's basically a word in RNA. I told you that we don't start translating at the 5' end. The ribosome sticks on the 5' end, but then it just glides. It's just scanning, looking for one particular trio, one particular codon that tells it start translating here. And that particular codon is AUG. And the remarkable thing is there are thousands there are nearly infinite number of species, infinite number of creatures, and all of them read their DNA and start translating at one codon, AUG. It is always, 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 you start translating at AUG. Now notice that's not the transcription start site. Transcription doesn't care about codons. Codons only matter in translation. You start at AUG. This is the start. It's known as the start codon because you always start at AUG. So the ribosome is going to come along here and scan. Boop, it's going to scan and says, no, 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 no. Oh, right here. Boop, boop, boop. There's AUG. And the first AUG I find, starting at the 5' end, I scan, boom, I hit AUG, I start there. Now, AUG, so we're going to bring the ribosome over here. I'm so sorry I'm all over the place today, guys. If you've been watching my videos, you know I'm all over the time. So hopefully you'll bear with me here. Okay, so the ribosome just glides along here, and it finds AUG and it stops there. Okay, now ribosomes are somewhat blind. So it kind of needs to know how to translate AUG into whatever amino acid is supposed to go there. Now notice, start codon still translates to an amino acid. It just also happens to be the start codon. So a lot of people see the start codon and then skip to the next one, but it's not. You read AUG and it puts in an amino acid. The way the ribosome knows how to put it in is there are a different kind of RNA called tRNA. There's the name tRNA. So this was the mRNA. That was the one that carried the gene to the outside or out to the ribosomes. The rRNA was part of the ribosome, and the tRNA, its job is to carry or is to deliver the correct amino acid to the ribosome to match a particular codon. The way I like to think of these guys, these tRNAs, is like a truck driver, right? You've got a delivery truck driver. He's driving. He arrives at the ribosome, and he's got a package in the back, right? He's got a specific package that he's going to drop off at the factory. And he looks at his clipboard and he says, this package is supposed to go here. Is this you? And the ribosome's like, oh, that's the right spot, right? So he compares so basically wherever the tRNA carries like a little stamp. It says, okay, this amino acid that I'm packing is supposed to be dropped off wherever I find an AUG on the mRNA. So then the ribosome pauses and says, okay, who's got the AUG? Gotcha, there you are. Grabs the tRNA and plugs him in. So it turns out that AUG codes for methionine. And the way it knows that is methionine's specific tRNA has a trio of letters called an anticodon. So remember, the codon is on the mRNA. An anticodon is a trio of letters on the tRNA that's complementary to the mRNA codon, right? Trio of letters on the tRNA that is complementary to the codon on the mRNA. And we're not going to make you interpret anticodons, but just to show you you're supposed to know what they are and that they are on the tRNA, not the mRNA. But we're never going to make you interpret anticodons, meaning, notice this tRNA, so this dark red thing, that's my tRNA. It has UAC as its anticodon, and that pairs to AUG, and that's how it knows that it's in the right spot. So the ribosome says, aha, the anticodon pairs up with the codon. That must mean that this amino acid that's being carried by that tRNA is supposed to go right here. That's how it knows. But for us, because we're human, we don't want to try and have to, or we don't have the whole collection of tRNAs there. We don't want to try and memorize all of the anticodons with what codons they go to. We're not a ribosome. So if we're going to try and figure out how to read the RNA and turn it into protein, we need a dictionary, because we don't have tRNAs to do the translation for us. We need a dictionary. So we have a table that looks something like this. There are various versions of it, but it looks basically like this. And the goal of this is our dictionary. It says this codon, now notice not the anticodon, this codon translates to that amino acid in protein, okay? So what this is doing is saying codon, this codon translates to this amino acid. That's what it's doing. So this allows you to play the part of the ribosome. And I'll show you how to read this table here. Basically, this table is designed to be easy, or to find an amino acid easily. Turns out there are 64 permutations, or there are 64 combinations of A, C, G, and U that you can put together in groups of 3. And it's not always logical which amino acid goes where. So we've structured this table to help you find whatever codon you're looking for quickly. So down the left-hand side of the table, we've listed the 4 codons. Down here on the left side, let's say we're looking for AUG, right? I've got this, I have the codon AUG. So I go to this first column. The first column here on the left side, this is where the first letter in the codon is found. So this is the first letter in the codon. So I'm going to find A. There's A right there. And then I go to the top. This out of the way. Then I go to the top. The second letter in the codon here is up here in the top. Okay, so the second letter in my codon is U. So there's my U. And then the third letter is down the right-hand side. So this one, my last letter is G. So I'm going to come down here, G. And what we're going to do is triangulate them. So I'm looking for A. So I go A over here, U up there, G, and I go to where they meet. Boom, there's AUG right there. Sorry, I should get a different color, huh? Okay, there's AUG right there. So that's not much better. So what this tells me is, so that's how you find a codon. If I'm trying to find, let's say, CAC, I'll find the C on the left, the A on the top, the C on the right, and triangulate them. Boom, there's CAC. So that's how you navigate around this table. And then what the table says is AUG translates to MET. MET is the abbreviation for methionine. I'll write that one out. That's probably one of the few amino acids you actually need to know its full name. Methionine. Not to be confused with methamphetamine, which is a drug commonly known as meth. Methionine, not methamphetamine. Okay, methionine is the amino acid that is encoded by AUG, which means every single protein starts with methionine. Every single protein starts with the amino acid methionine. Every single one, because because AUG is the universal start codon. So you always start with methionine. Okay, so the way we go about translation here is we found AUG, so we can now that I showed you the concept of the anticodons, let me delete them so that they don't confuse you, because we care about codons, not anticodons, but know that they exist and what they do. Okay, so we've read AUG, we stuck that there. Now ribosome, the whole ribosome packs up and shifts over one. And now we're focusing on this trio. So notice we're not shifting over one letter, right? We're not now reading that as the next codon. AUG is one word. Now we go to the next trio of letters, completely next one. So we're talking about CAU, not UGC. AUG is one word, stop. we move to the next one, next word. So CAU, we're going to come to our table over here. There is a C here on the left, an A up here on the top, a U on here on the right. Go over here. CAU translates to HIS, which is histidine. But you don't need to know that. So you'll just find HIS, and it sticks it right there. And then the ribosome will actually glue the methionine to the histidine. So it actually breaks the methionine off of the tRNA and glues it to the histidine. And then this tRNA floats away. And then there are other enzymes that will reload a new methionine on top of it so it can be reused. Kind of fun, huh? So now I just linked my first two amino acids together. We're starting to build the protein chain here. And then the ribosome will shift over. To the next one. Okay, and we read CGG. And we're going to come over here. And you go CGG. Right there. Translates to ARG, which is arginine, which again, you don't need to know. So we're going to take arginine and bring it up here. And then we're going to take histidine and stick it to the arginine, break it off of this tRNA, and this one's going to float away. And then we're going to shift the ribosome over again, et cetera, et cetera. AUC will come over here to the table. We find the A, U, C. There it is. Now, this one trips people up. And I always want to, and I want to make a point of this because not that it's super duper important, but if it just to help avoid confusion here, whatever moron decided what the typed English language was going to look like, decided that the capital letter I was going to look exactly like a lowercase l. And someone should figure out who did that and like lock them in jail because it's so stinking frustrating. A capital I looks like that, right? So this is actually not LLE. It is ILE. Okay, that stands for isoleucine. It's ILE, not LLE. So again, you wouldn't get you'll never have to like fully write this thing out, but just remember, just don't be confused. That's not two L's and an E. That's an I, an L, and an E. Okay, there's my little pet peeve right there. All right, sorry, I am all over the place today, guys. Thanks for putting up with me. I'm sorry this video is so long. Okay, so we have so now we have methionine, or so we okay, so we have isoleucine. So the AUC translated to isoleucine. So let's come back over here and we will find our isoleucine. There it is. And we will stick it in right there. And then we will connect the arginine to the isoleucine, break it off of here, and the tRNA floats away. Okay, now we move to the next one. Let's see here, trying to just decide how far to go. Okay, we'll do 3 more. Okay, because this is, because I am getting to a point here. So UAU, that's tyrosine. I'll just do these 2 here quick. UAU is tyrosine. Get this guy out of the way here. We move over. UGC, where'd it go? UGC cysteine. Okay, now here's where things get interesting. Because I know you're probably bored out of your mind and you're regretting watching this video, and for that I am sorry. Okay, that floats away. Now we shift over to this codon over here, and we read UAA. we come over to our table, UAA, stop. Whoa now, that's not an amino acid. This codon, notice up here in this corner here, there are 3 codons that don't code for an amino acid. They code for stop. Remember with transcription, we had a specified transcription stop site. Here, we know when to stop building the protein because we hit one of the stop codons. UAA, UAG, and UGA are the 3 codons that say stop. And what really happened, or the reason why they say stop, is there isn't a tRNA that has an anticodon that pairs to those. It physically doesn't exist. So there are 64 possible codons, but only 61 different tRNAs. 3 of the codons simply don't have a tRNA, and the ribosome is extremely OCD. So it comes along here and it hits UAA and it says, okay, tRNA, where are you? And it just chills there for a little bit. Nobody shows up, but it's not going to go to the next one because it needs to put this one in here now before we move on. So it's just going to sit there. Nobody comes because it doesn't exist. There isn't a tRNA for UAA. So it eventually just quits and the whole ribosome falls apart, floats away. And that's it, we're done. So notice, even though GCU over here is a completely valid codon, it would translate to, let's see, GCU would translate to alanine. I do not add an alanine because it's after the stop codon. And stop says stop. So when you're translating, you go through and you stop at UAA. Now, a lot of people make the mistake of coming in here, they're writing their amino acid sequence. So they come in here and they would write like MET, right? And then you connect that to histidine, and you connect that to arginine, and you connect that to isoleucine, you connect that to tyrosine, and you connect that to cysteine, and you connect that to stop, and you connect that to alanine, ... That's not right. That's incorrect. Stop is not an amino acid. Stop is an action. So you come in here and we don't add the alanine, and we don't even write stop because stop isn't an amino acid. It's not part of the chain. So the last amino acid in the chain is actually cysteine because it was the one before the stop codon, and stop is not an amino acid. So don't make the mistake of writing stop at the end of your chain because you might lose points on your homework if the TAs are in a bad mood. So you just write to whatever the last, when you're doing translation on your homework, make sure that you start at AUG, and then you write each amino acid as you translate them over. And then when you hit the stop codon, we just don't write anything. So the last one in the sequence is going to be the last amino acid is going to be from the codon before the stop codon. And then you never, ever, ever translate anything after the stop codon, no matter how valid the codon is. Okay? So then the ribosome will break up and fall away. And this chain of amino acids here floats away and is destined to become a protein. Now, I told you over here that translation is going from RNA to protein, but I told you that was the general idea. In reality, this is not yet a protein. Just a straight chain of amino acids is not yet a protein, and this is a vocab difference that frustrates a lot of people and confuses a lot of people. And I actually have an article about it on my website. Let me go reference those pictures. Clear my pictures, or clear my drawings. Okay. Let me go reference my pictures there because I think, because this is a very, very good point to make. What we built here in translation is what's called a polypeptide. It's not a protein yet. It's a polypeptide. The reason why there's a reason we call it that, and if you'd like to know, you can read the article. But a polypeptide is a chain of linked amino acids. That's what a polypeptide is. But a polypeptide is completely worthless. It's like a piece of paper. I love thinking of this like a piece of paper. When a piece of paper comes off the mill, it has absolutely no function whatsoever. It's blank, it's boring, it can't do anything. It's completely worthless. A piece of paper only gains function, only gains abilities if you fold it, right? If I take a piece of paper, let me come in here and draw here, because I think this is a very important concept to know. If you take a piece of paper and I fold it into a particular shape, I grant it the ability of flight. If I fold that piece of paper into a different shape, I grant it the ability to float in water. But the piece of paper in and of itself has no actual function, but folding it is where it gains its function. And the particular shape in which I fold it, the three-dimensional shape of the piece of paper, is what confers its abilities on the piece of paper. So same thing with a polypeptide chain. A polypeptide chain can't do anything. But once it's built, it actually automatically folds in a specific three-dimensional shape. It's like a paper airplane that folds itself. It's awesome. It folds itself into a particular shape, and the three-dimensional shape allows it to do its job. Once it gains its three-dimensional shape, now we call it a protein. So a protein is a polypeptide that has been folded into its three-dimensional shape. That's the difference. So translation, we technically build a polypeptide, but it instantly folds itself to make a protein. So we just summarize it as saying translation takes you from RNA to protein because it's basically the same step. But there's a good distinction to make, because in translation, we build a polypeptide and then the polypeptide forms into a protein. And this leads us into the next part of our discussion here on mutations. Because in your homework, we ask you about the consequences of mutation. And there are, what we mean by a mutation is a change in the DNA or RNA sequence for a specific gene, an alteration. And there are a bunch of different mutations that could possibly happen. In this class, we're going to focus on there's one category of three particular mutations. All we're focusing on this class is what happens if I accidentally swap out one base for a different base in the DNA. That's what we're worried about in this class. So let me refresh this. We'll bring up a brand new version here. Brand new version of my simulator. And we're going to talk about the three different kinds of mutations you can have and how you can tell which one's which, and what the difference between them all are and what the consequences of all of them are. Mutations can happen in all kinds of ways. You could get a chemical called a mutagen that can change your DNA. You know, there's a lot of chemicals that say, don't get this on your skin, because it could do that. You could have a sun ray come in there and accidentally knock a base out, and then the cell puts in the wrong one. So that's why they say if you get sunburns, you could potentially get cancer. That's why they're worried about skin cancer, because if you mutate the DNA in the wrong way, you could actually get cancer. And the sun has a powerful ability to be able to occasionally mutate the DNA. So that's how that happens. So there are lots of different ways to mutate the DNA, but we're just assuming it's been mutated. And so let's say here, for example, okay, let's assume just for an example, let's say that this little base pair right here was changed to, let's make this one I don't know, let's make it into a G, which would make it paired with a C, okay? Let's say we accidentally came in and switched that. So this is a mutation. This is not what the original gene was supposed to look like. This is a mutation. So something can only be a mutation in comparison to an original, right? You can only be different if there's something that you're comparing to. So, so a mutation is always compared to the original. So the original had a G in that spot, and I put a C there instead. That was the change that I made. So let's see what the consequences are. We're going to go through transcription, and we're going to come out with a strand, or with an mRNA strand. I'm going to go ahead and speed that up here. Okay, I went ahead and sped this up here for sake of time. So I went in and did the transcription. So I read the template strand, and I transcribed it into an mRNA. Now we'll come down here and we'll do translation on it. Okay, so here's our new mRNA, our modified mRNA. So now we're going to do translation. So ribosome is again so we're going to scan along, and I won't bother to move the ribosome for sake of time, we're going to go through and we're going to translate it. So we scan along, find the first AUG. There it is, AUG. And that always translates to methionine. Let me take out these anticodons again. So methionine, we're going to stick in methionine, because we know that's always the start codon, and methionine always goes with AUG. So AUG pairs with methionine. Okay, now we're going to look at the next one, GAU. We come over here to our translation table. You can just Google this to find it, or you can get it from my website. I have it down here in the image section of the week 5 page, so you can get it there. But the Google ones are fine. On the exam, we will give you one of these, so you do not need to memorize this table. The whole goal is for you to get the point, not to memorize a bunch of useless information that you'll never use in your major. Like I'm a bio major, and I didn't even have to memorize this. So don't worry about memorizing the table. You do not need to memorize the table. Just know how to use it. Okay. So we put in, so AUG was the start codon. We put in methionine. Okay, now we got GAU. So let's come to our table. GAU is ASP. Oh wait, that's a problem. Because remember, well, actually, let's finish this up first, and then I'll convey the point. Okay, ASP goes right here. And I won't bother removing the tRNAs again for sake of time, just remember. So now we're at CGG. We come back over here. CGG is arginine. And then we go to AUC, which is, let's see, AUC. There's our isoleucine. UAU. There's tyrosine. UGC. UGC is cysteine. And UAA is a stop codon. So we stop there. Okay. So now here's our amino acid sequence with the mutated DNA. We've got methionine, aspartic acid, arginine, isoleucine, tyrosine, and cysteine. Again, you don't need to know that. You can just write down their three-letter abbreviations. But what we had before, remember, this is in comparison to the original. So on your homework, question 2, we have you transcribe and translate a DNA sequence. In question 3, we took that DNA sequence from question 2 and we mutated it in a couple of different ways. And so when you come out with your answers on question 3, it's always in relation to question 2's amino acid sequence, which was the original. So just know that. That's another question on the homework that a lot of people get confused on. So in this case, the original strand, or the original amino acid strand that we did just a few minutes ago, if I copy this down, that wasn't aspartic acid because it was CAU before. The original strand was CAU. We had a histidine right there. The original strand was histidine. So let me write out original sequence. Thank you for putting up with me, guys. Okay, here we go. Original sequence. So this is what it was. And now we get to compare this strand up here to this one. If you notice the difference, this second amino acid is completely different. By changing one letter, I put in the wrong amino acid. Now that's usually very bad. Occasionally it can confer a benefit and it's like, ooh, and thus evolution dominates, right? But most of the time that's really bad because we told you that proteins gain their jobs are able to do things based on their 3-dimensional structure and that they self-fold. Well, their 3-dimensional structure, how they self-fold, is determined by what amino acids it has in what order. So if you put in the wrong amino acid, it could potentially cause the protein to fold wrong and completely make the protein useless. It will not do its job. Or worst case scenario, it could actually do the wrong job and be harmful to the cell. There are cases where that happens. If you've heard of mad cow's disease or scrapie, or things like that, though that is that's a problem where you had a single, or where a protein folds wrong and actually can kill you in the process. So we don't want our proteins to fold wrong. That's a worst case scenario, but we don't want our proteins to fold wrong because that is a perfect way to get diseases. In week 9, we'll learn sickle cell anemia is caused by a single base pair mutation, and yet it causes their blood vessel or their blood cells to completely change shape, not be able to hold oxygen as well, get stuck in their blood veins, and it causes extreme pain and is a miserable way to live. One base pair can change, or a 1-letter change in the DNA can change one amino acid and a protein and completely mess the whole thing up. That's why these mutations are really, 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 really bad. And that's why we try to avoid mutating the DNA as often as we possibly can. So in this case, this particular mutation, I just swapped out an amino acid. Notice it's still, the polypeptide chain that I made, the mutated polypeptide chain that I made, is still the same length as the original, right? It's still 6 amino acids long. But I swapped out an amino acid for a different one. This kind of mutation is called a missense mutation. Let me write that out for you. Okay, this particular mutation is called a missense mutation. A missense mutation is any mutation where I simply swap out one amino acid for a different one. So we swap one amino acid for a different one. So this is an example here of a missense mutation because I mutated the DNA and that caused me to put in the wrong amino acid. So another way of saying this is it's a mutation that causes us to put in the wrong amino acid. So this is a missense mutation because I put in an aspartic acid instead of a histidine at this second spot here. That's bad. And the way that I remember that missense is a missense mutation is when you swap out the wrong amino acid, or where you put in the wrong amino acid, it's a mistake. So we went through and we put in all of the ingredients, but I accidentally swapped out sugar for salt, and my cookies are a disaster and an insult to all mankind. So that's a missense mutation, is I went in and made a mistake and put in the wrong amino acid while I was going in. That's a missense mutation. The second kind of mutation is, what if I mutate the DNA? So let me revert this back to its original, and we'll just pretend we did transcription already. So pretend I've mutated the DNA and it's going to have this effect. So the original here, let me change it back to how it was. So this was the original. That was the original mRNA sequence. What if we mutate this to a G? This one, notice, if I read AUG, I will get methionine. CAU, that will be our histidine friend. CGG still codes for arginine. AUC still codes for isoleucine. UAU still codes for tyrosine. UGC still codes for cysteine. And UAA still codes for a stop codon. Look at this. If we compare this sequence to this one, they're identical. What the heck? I mutated the DNA and yet nothing happened. That's because we mutated the DNA before the start codon. And because we just scan over that, the mutation is not going to have any effect. It's basically like mutating in the junk DNA regions. It's a mutation, yes, because I still did swap out a DNA base and it changed the mRNA, but it had absolutely no effect on the end result protein. So this is called a silent mutation. A silent mutation is one where I change the DNA, but don't change the protein. So we change DNA. So I mutated the DNA sequence, but it had no effect on protein. No change in protein. So that's one way I could do it. There's actually two ways you can get a silent mutation. I changed that U to a G right there, which was before the start codon. So if you have a mutation before the start codon, then it's going to be a silent mutation every time, because it's not going to affect the protein because we don't read anything before the start codon. Or after the stop codon, if I were to mutate this C into something else, it's after the stop codon, which was right there. So again, it would be a silent mutation because it wouldn't affect the protein at all. Does that make sense? So in this case, silent mutations are completely harmless. They're not going to ruin the protein at all. The proteins will still function exactly the same. You won't be able to even tell you have a silent mutation. Okay? So if you've got, so one way is you can have the mutation before the start codon or after the stop codon. Or there's another way you could do it. And this one is really fun. So this is also going to be an example of a silent mutation. I'm just going to erase this so that we can change screens here. What if, and let me look up an example here. Okay, what if I change, this G to a U? that looks like a missense mutation, right? I changed the base, which is going to change the codon, which means which means I expect it to translate for a different amino acid, and it's going to be a missense mutation. Let's go look. So AUG still codes for methionine. AUG. We know that CAU is still going to code for histidine. We didn't ruin that one at all. This is the codon that we messed up. It used to be CGG, which coded for arginine. Let's go look up CGU. What does that code for? CGU. Oh, look, arginine. So I will stick in an arginine and then the isoleucine, tyrosine, cysteine. Boom, it's the exact same protein. Well, what the heck happened? I changed a codon. It wasn't before the start codon. It wasn't after the stop codon. I changed a codon. So how did I put an arginine there? How did this turn into a silent mutation instead of a missense mutation? Here's why. Remember, I told you there's 20 amino acids, but there's 64 possible combinations of 4 letters combined into groups of 3. There are 64 possible codons, but only 20 amino acids, which means more than one codon can code for a certain amino acid. If we go look at our table, look at this. Leucine has 6 different codons that all code for leucine. Right? It's like in the English language, we've got like synonyms for things, so you can have like 40 different ways of saying the same exact term. There's 6 different codons that all mean leucine. Over here, arginine has 6. In fact, all of the amino acids but 2 have more than one codon that can code for them. AUG is the only one that codes for methionine, and UGG is the only one that codes for tryptophan. All of the other amino acids have more than one possible codon. So if I mutate a codon where it switches from one codon to another codon that still codes for the same amino acid, it again has no effect on the protein, hence it's known as a silent mutation as opposed to a missense mutation, which was where we swapped out an amino acid. So a silent mutation by definition is one that has no effect on the protein, no matter where we mutated. A silent mutation is one that has no effect on the polypeptide sequence. Okay, so in this case, that mutation was a silent mutation because I still put in the same amino acid as I did before. Good so far? So there's our first 2 types of mutation. The missense was we put in the wrong base. A silent is nothing happens to the protein. And the third one is an interesting predicament. Let me come in here. So let's reset our DNA sequence back to where it was. So let me fix my, okay. So this is where it was. And this was a CAU. Okay, there we go. There's the original sequence. What if I come in here and mutate, let's see. What if I mutate? Okay, what if we mutate, oops, yeah, this one right here. What if we mutate that one to be a G? So let's change it over here, UAG. Okay, what does that do? So we didn't touch AUG, so I know that one's the same. You can save yourself a lot of time by, once you figure out the original peptide sequence on question 2 for your homework, you can copy that one and paste it on each of the 3 mutation ones, and then just go look for which codon did you alter and see what it did to that particular amino acid, and just change that one. It'll save you a lot of time. There's your little trick of the trade there. Okay, so So we changed this one from UAU into UAG. So I know that everything before it's going to be the same because I didn't change any of these codons. So the only one I'm worried about is this codon right here. Let's see if it still codes for tyrosine. So it's now UAG, UAG, right there. That's a stop codon. Notice I turned it from tyrosine into a stop codon. What does that do? We came down here, that's a stop codon. That means the ribosome won't put a tyrosine right here. And because we never translate anything after a stop codon, that means it won't put the cysteine there either. So we stop. I put in the 4 amino acids and that's it. That is what is called a nonsense mutation. Write that out. A nonsense mutation. A nonsense mutation is any mutation that results in what they term a premature stop codon. Basically, if I stop before I'm supposed to, that's a nonsense mutation. So the mutation changes a codon into a stop codon. An early stop codon. And these are really bad. This is probably the worst of the three because a missense mutation, yeah, we can sometimes get the protein to still work if you have a missense mutation, depending on where the mutation is and which amino acid it switches out for. Occasionally, your body can still make a missense mutated protein work. A nonsense mutation, that's physically impossible. You can't do it because we're simply missing half of the story here. So basically, it's like reading a recipe and only getting down or putting in all the dry ingredients and then throwing it in the oven. You're just going to come out with burnt flour, right? I didn't add the milk, I didn't add the eggs or anything. If I just forget to put in half the ingredients, it is not going to work. And proteins work the same way. If you just don't put in the rest of the polypeptide, it's going to ruin the whole contraption. So nonsense mutations will completely make a protein worthless. It'll completely make the gene useless. The instructions won't make a functioning protein at all because it'll make it halfway through and just quit before it's done. And the protein's not going to fold right. It's not going to do its job. And we just wasted our time and energy and could potentially cause a lot of problems in the cell, depending on what the protein's job was that I'm not building. So that's a nonsense mutation. So just to review, a missense mutation is when you change a base in the DNA that causes you to put in the wrong amino acid in a protein sequence, but the protein sequence is still the same length. I'm just swapping out the one amino acid for a different one. That's a missense mutation because I'm making a mistake. A silent mutation is when I swap out a DNA base that causes no effect on the eventual polypeptide sequence, no effect on the protein. And the three ways you could do that were have it be mutated before the start codon, be mutated after the stop codon, or change a codon to a different codon that still codes for the same amino acid. All three of those are silent mutations. And it's silent because you don't notice it. Does that make sense? That's why it's called a silent mutation, because it has absolutely no effect. So it's quiet. The last one is a nonsense mutation. And a nonsense mutation is you stop early. That's anyone where you change a codon into a stop codon and stop the protein before it was supposed to be done. You wind up with a shorter polypeptide than you were supposed to have That's a nonsense mutation. I've got a couple of ways of remembering that. One is the, like, if when teenage girls are arguing with their mother and they're just screaming, they're bawling their eyes out, and they got this big, huge explanation and whatnot, and the mom's just like, stop, I don't want to hear any of this nonsense. You stop the conversation early. Don't care what the daughter has to say. We're stopping it early. That's a nonsense mutation. We want to, when we're stopping early, it's nonsense. Another way you can remember that is if you've ever had somebody like trying to tell you something and then they just kind of drift off in the middle of their sentence and don't complete it, it never actually makes any sense and you're just left there confused. Happens to me all the time. So it doesn't make any sense. Like whereas if you were to just swap out one word, you could probably be a little confused, but you could still make out the meaning. If you just stop the sentence early, we're just left in suspense. So it's a nonsense mutation is you stop early. So your goal on the last question of your homework is to go through and say, well, I changed this, I changed that, I changed that. What do these changes do to the protein? And then you have to determine whether it's a missense, a silent, or a nonsense mutation. And that's it. Okay? So I am so sorry that this video was so dang long, but hopefully you were able to make it through it and find some helpful information. If you have questions, want some things clarified, please feel free to reach out to me or your instructors or your TAs. Set up a tutoring appointment with me. We can talk it through, clarify your questions. But hopefully this video helps make the readings make a little more sense and will help you on your homework as well. So thanks for watching and have a wonderful day.